Welcome to the Pesticide Resistance Management course. Pesticide resistance occurs when pest populations change genetically so that they are less susceptible to a pesticide or to a class of pesticides. We see resistance occurring among pathogens, within insect populations, and in weeds. In this course, we'll take a look at resistance in each of these disciplines. This course is divided into 3 narrated presentations: the first on fungicide resistance, a second on insecticide resistance, and finally a third on herbicide resistance. Although pesticide resistance in weeds, insects, and pathogens may develop by similar mechanisms due to high selection pressure and high genetic variability within pest populations, there are some differences in how to manage resistance within the different disciplines. The objective of this course is to highlight how pest resistance develops and how we can delay and manage resistance to fungicides, insecticides, and herbicides. In this first section of the course, we'll be focusing on fungicide resistance. We'll talk about various fungicides, including what is available in terms of different classes and modes of action. Then we'll take a look at some examples. We'll be focusing on foliar pathogens. Foliar pathogens usually all act about the same in terms of epidemiology. There are certain epidemiological parameters that drive the disease and pathogen biology that are similar. Most need moisture to cause disease. They also need high humidity for sporulation. Foliar pathogens are polycyclic. There are many generations during the growing season, or at least the potential to have many generations. When we talk about fungicide resistance, we don't really include the root rots. With root rots, you may have a fungus that infects a certain root this year and the next year will go infect another type of root. So there aren't the same disease dynamics with soil-borne pathogens as with aerial pathogens. Let's start out by taking a brief look at some of the different fungicides, and then we'll go into a little more detail. We have the SBI types, also called the DMIs. The DMIs are demethylation inhibitors. There are some differences in chemistry, but they all attack the fungus in the same locus. Some examples are Raleigh, Vidicure, Elite, Vintage, Balaton, Orbit, and a number of other materials. This group works well against powdery mildew, leaf spots, and fruit rots. If there is high disease pressure and you use these materials over and over, there's a good chance you're going to get some resistance development. Vidicure is the only fungicide in this class that has multiple sites of action. It was documented by the University of Maryland that this product worked when other DMI fungicides didn't. We've seen this in grapevines for grape powdery mildew. Even with sublethal doses of this product, we haven't been able to develop further resistance. A completely different and newer class of fungicides is the strobilurins. Some examples are Sovereign, Flint, and Abound. These attack any fungus in a different location than the SBIs. If resistance develops to this class, we will have basically lost all of these products because the whole population would have shifted over to resistance. The quinolins. The only fungicide we have in this group is Quintec. It's got its own class number and it attacks the fungus in a different location than the others. There are also combination materials such as Pristine, Adamant, and Inspire Super. These are combinations of DMI fungicides and strobilurin fungicides. There are some new chemistries being developed. A new class of products that we have available is SDHI. These are really super fungicides. Bayer's product is Luna. BASF has one called Maravon. Other companies are now working to bring these products out as well. It can cost between $50 and $100 million to get these chemicals registered for use. Let's go into a little more detail now about the groups of fungicides. We'll start with the benzimidazoles. Benzimidazoles were introduced in 1974. These are locally systemic. Benlate by DuPont was one example, but was pulled from the market a few years ago due to lawsuits. It can still be used in other countries, but it wasn't worth keeping in the U.S. Topsin M is a similar product, but when applied, it doesn't need to be broken down into its active ingredient. It goes to work immediately. The benzimidazoles are members of FRAC Group 1. FRAC stands for Fungicide Resistance Action Committee. It's made up of members of every company who has a fungicide in that particular group. So DuPont and Surexagri are the only members of FRAC Group 1. They get together and talk about ways to mitigate resistance that has developed. They might meet once a year or every couple of years. The mode of action for frat group 1 fungicides is that they work as a single site inhibitor that interferes with nuclear division in the fungus. The benzimidazoles inhibit mycelial growth and sporulation. Topsin M is registered in grapes for canker disease control. It is a dormant over-the-vine spray in combination with Raleigh, which is a DMI fungicide. The resistance risk is very high, so resistant populations do not decline in absence of fungicide use. In the presence of resistance, if you use this product one time, the entire population shifts into completely resistant mode, and you just can't use it again that year. If you can wait a year, there's some reversion back toward a wild type, but it never goes all the way back, so you can probably use it again once the next year. If it is used as a dormant spray, you probably aren't putting a lot of selection pressure on the pathogen. If you put it on when the pathogen is sporulating, then that's different. It's recommended that we use Topsin N for canker diseases in combination with a product of different chemistry. Let's move to the demethylation inhibitors, often called the DMIs or SBIs. Several products in this group have the same chemistry. They are triazoles. The first example is Balaton. This has been commercially available in California since 1982. Its active ingredient is triadimephon. This is locally systemic. Elite is a newer DMI fungicide. We would expect to see cross-resistance. Other examples are Metal with the active ingredient tetraconazole, Indar with the active ingredient fenbuconazole, and Orbit with the active ingredient propraconazole. Orbit is registered on tree fruit and some vegetables. Bumper is another with the active ingredient propraconazole. Raleigh with active ingredient myclobutanil, and Inspire with the active ingredient difenconazole or other triazoles. Procure with the active ingredient tyflumazole is an imidazole. It's interesting when we look at Procure and cross-resistance with triazoles, we don't see high levels of resistance. Rubigan with the active ingredient pheneramyl is a pyrimidine, and this is the other one where we don't see cross-resistance development. So in the triazole class, we would expect to see a little loss of efficacy under high population dynamics. The SBIs belong to frat group 3. The mode of action is single-site inhibition. They inhibit demethylation and other processes in sterile biosynthesis, and they are locally systemic. This group of fungicides will inhibit spore germ tomb elongation and therefore shut the fungus down. We also see mycelial growth inhibition and suppression of sporulation. Some may say this group of products are eradicants. That's probably the worst way you could use this group of fungicides. They are preventives. There is resistance in California to this group of product. The quinolines include Quintec with active ingredient quinoxaphene made by Dow AgroSciences. This is a contact fungicide, not systemic. The quinolines are in frat group 13. The mode of action is probably a single-site inhibitor, but it disrupts early cell signaling. It suppresses spore germination, early germ tube development, and/or aprosorium formation. There is no effect on sporulation. The resistance risk is now considered to be medium. The only fungus it attacks is powdery mildew, and there wasn't a reason to bring this product out when there were all the DMIs and other products that were labeled for use. When we found resistance in the DMIs, this product was brought out primarily to combat resistance in the DMIs. It has worked beautifully for a number of years now. It's a good material. Some say it's not working as well as it used to, but that's true of almost all fungicides and probably all pesticides. As you use them, selection pressure gets stronger toward the fungus. It's probably not going to work quite as well. So you lose a little bit of sensitivity in the population. Within the strobilurin group, there are a number of products. These are both contact and systemic products. Abound from Syngenta, Cabrillo from BASF. Evito is a product mostly used on tree fruit, but we don't have it in California. Flint is from Bayer. Pristine from BASF is mostly used in viticulture and has been used more against powdery mildew than botrytis. We're starting to see some problems now. Sovereign is also by BASF. The mode of action for this group, FRAC 11, is single-site and blocks respiration by interfering with the cytochrome B complex. This is a different mode of action from other classes. Strobilurins inhibit spore germination and mycelial growth. There is no effect on sporulation. The risk of resistance is high. Each one of the classes of chemistries that we've looked at so far attacks the fungus at a different loci, and that is why it is best to rotate products, before resistance develops, and especially after. Next, we have the combination products. Pristine is the first example. It's a combination of pyraclostrobin and boscalid from BASF. The mode of action is both contact and systemic. In this particular combination prepackaged mix, it is a group 11 and group 7. The growth effects are the same as for the strobilurins. They are unknown for carboxyanilid. The sporulation is the same for strobilurin and unknown for carboxyanilid. The resistance risk for this is low to medium. Pristine is mostly used against powdery mildew, but it has been used in grapevines for both mildew and botrytis due to its combination. The powdery mildew rate is listed as a low rate, and the botrytis rate is listed as a high rate. We've now found 3 locations in California where botrytis populations are highly resistant to boscalid. If you use this product over and over and over again in a vineyard or strawberry field for powdery mildew control in the presence of botrytis, you're applying selection pressure against botrytis by using the low mildew rate. Over time, resistance develops in the non-target organism, botrytis. You can still use boscalid if you use it at the high rate for botrytis. If you are going after mildew, you can still use the low rate, but switch to something else for botrytis. How do fungicide rates play a role in fungicide resistance? Not too long ago, it was recommended to use low rates because it was thought that low rates would allow resistance development as a slower rate. We now know that full rates should be used in every application. Low rates are still being used in prepackaged mixes. The rate for one product in the mix is reduced to make the product cheaper. The bottom line is if you are using lower rates in mixes, one of those products is going to last longer than the other one, and there are members of the fungus population that have differential sensitivities to products. The sensitivity over on the right side of the population where we start seeing resistance can attack the fungicide concentration as it decays over time at an earlier date than the population on the left. So right away we are putting selection pressure on the part of the population that we don't want to put selection pressure on by using lower rates. For the succinate dehydrogenase inhibitors, the SDHIs, there are 3 products. Endura is one. The active ingredient is boscalid. Penthiopyrad is DuPont's new material. However, they are going to limit the number of crops it's used on. Luna Privilege of the Luna Group is the 3rd product. The active ingredient is fluopyrim. These are all single-site action materials. The resistant risk is high. There is still quite a bit we don't know about this group, but they work well against foliar diseases. So what is resistance? It can be defined as a change in the sensitivity of a pest population to a particular product, a pesticide, resulting in the failure of a correct application of the pesticide to control the pest. This definition can apply for all 3 disciplines: fungicides, insecticides, and herbicides. Resistance can develop when the same pesticide or similar ones belonging to the same class with the same mode of action are used over and over again. There are 3 types of resistance: tolerance, where continued use over time may allow the fungus to develop some sort of tolerance to the product; reduced sensitivity, like we see with the DMI fungicides; and complete resistance, like we see with the benzimidazoles and strobilurins. There are several ways that populations of fungi can become resistant to fungicides. One of these is the altered target site. So think of a fungicide that has a specific site in the fungus where it attacks to kill the organism, or where it acts to disrupt a particular biochemical process or function that will result in death. If this target site is altered, the fungicide can no longer fit in the groove. It's kind of like putting a square block into a round hole. If the fungus has the ability to alter the site where the fungicide normally attacks, then the fungicide is blocked from going into that site and killing the organism. This is the most common mechanism that fungi use to become resistant. Detoxification is another way fungi become resistant. The metabolism within a fungal cell is one mechanism a disease pathogen uses to detoxify a foreign compound such as a fungicide. The fungus has the ability to quickly degrade a fungicide and potentially inactivate it before it can reach its site of action. A third way fungi become resistant is by reduced uptake of the fungicide. This occurs when the resistant pathogen simply absorbs the fungicide much more slowly than the susceptible or sensitive isolate. Finally, there is removal or efflux. A fungal cell may rapidly export the fungicide or move it somewhere else before it can reach the target site of action. Now we're going to spend a little time discussing a problem we ran into with strawberries a few years ago. We found that all of a sudden a new fungicide, Fungicide, Elevate, wasn't working as well to control botrytis fruit rot. If we look at the disease data from the field trials, for the untreated controls you have about 32% disease. With Elevate, it knocked disease down to 23%. Not until you get to switch do you really start seeing a difference in the degree of control that is desired. In 2006, we had the same kind of data, but where we used Elevate, we had a pretty high degree of disease. When you've got a resistant population with some members being less resistant than others, and you apply a fungicide, you knock out the sensitive part of the population. But if we still have high selection pressure with rainfall in the case of botrytis, the resistant part of the population continues to increase at a more rapid rate. Usually by the second or third application, you've lost control. To manage resistance in botrytis cinerea, we need to avoid consecutive applications of one fungicide or fungicides of the same chemical group. Use multiple fungicide rotations of premixes of products, such as Captivate, Pristine, or Switch, to keep the fungicide pressure low. Finally, it's important to monitor the insensitive populations. We can prevent and manage resistance development by rotation and by mixing classes of chemistry. Luckily, Fungicide resistance, the very highly resistant population, dies out over the winter. However, after 2 applications, we flip it back into high resistance. So we can still use the product, we just need to know what's going on with resistance and with sensitivity of the population. Let's take a look at DMI resistance now. It is relatively easy in California to find field or lab mutants that are resistant to DMI fungicides. It suggests that a rapid resistance would develop in the field. This has occurred. When Balaton was introduced, it was thought that resistance would be less problematic because of the way the fungicide attacks. It was thought that one application would not give resistance, but that over time, you would get a directional shift. It was thought that resistant isolates would be less fit than wild types, but that didn't turn out to be true. However, we do have management strategies in place. If you look at a typical population, anything on the right of the center line would have some resistance in it. When Balaton was introduced, this is what we saw. It was very effective against the fungus. Reduced rates were being used and resistance did develop. We found members of populations that were not killed at 3.5 ppm. Eventually in 1986, just 4 years after the introduction, this part of the population could attack a 4-ounce application of Balaton 6 hours after it was applied. If you're spraying a product to knock out the sensitive part of the population, then you have a highly resistant part of the population that keeps getting more resistant with subsequent applications of the same chemistry. By about 3 to 4 applications, we're dealing with a population that's completely uncontrollable with that product. It doesn't mean that it can't be used, you just have to know what you're dealing with and use it in the right way. We documented that over the course of the season, the 3 DMI fungicides that we had available to us all increased in resistance. With Rally or myclobutanil, there was a 10-fold less degree of resistance than with Rubigan or Feneramol, which was probably about a 100-fold decrease in the degree of resistance. We found that temperatures actually mask the presence of resistance. If it is high enough to kill the fungus by itself, then fungicides aren't necessary. So here is isolate REN1, a wild-type isolate you can see over the course of 50 days or so. The lesion slowly dies out. With a 4-ounce application of Balaton, you get immediate kill. For isolate LO2, a highly resistant population, the control over time slowly dies out. With the resistant population, an application of Balaton at 4 ounce knocked it down, but then it came back. A second 4-ounce application knocked it down again, and eventually after 3 applications, we killed the fungus. So at temperatures under 25 degrees, which is optimum for the pathogen, it took 3 Balaton applications, or 12 ounces, to finally get control. At 32 degrees, which is at the bottom edge of negative growth for the fungus, you can see that 1 application of Balaton killed the wild type, but the temperature itself also killed the wild type. With LO2, with high resistance, you get beautiful control with high temperatures. So in the years we saw pretty severe disease, it was because we had pretty moderate temperatures and very high degree of pathogen reproduction. The populations of spores in the field are very high. So high temperatures mask the presence of resistance. We redid this study in 2002. The black bars are the original EC50 values, and white bars are the populations we found in 2002. In every production area in California, we have increased the level of resistance to DMI fungicides, even though Balaton had never been used. The other DMIs had been used like Rally. Does resistance overwinter in Cleistothecia? And are resistance genes fit in nature? We extracted ascospores from grapevine bark in the lab, tested against Balaton, and found average EC50 values in our population of 22 ppm with a range of 2.27 to 49.34 ppm. After one application of Balaton, we saw about a 33 ppm average for the population with a very significant range. After 2 applications, 3 applications, and 4 applications, we increased the level of resistance in the vineyard. Then in October, we went back and collected leaves, extracted the ascospores, and retested them for resistance, and found we have reached EC50 of 33 ppm. This really surprised us because usually resistant propagules are less fit. But because they're being carried over in the fruiting body of the fungus, this means that this year in the spring, when these are released and cause disease, the grower is going to be dealing with that degree of resistance. Over time, we see that degree of resistance increasing. Resistance overwinters in the fruiting body, and this documents why we're not seeing a reversion back to the wild type in the population. As we conclude the presentation on fungicide resistance, it is important to take away the following key points. Most new fungicides are single site of action products and should never be used alone. Always use different classes of chemistry for rotation programs. Use broad-spectrum products when possible. Finally, remember that selection pressure leads to resistance.